Great, thank you. Hello, everyone. We're very excited today to have Barb Munch, who is the principal from Munch GLP Consulting. Just a little background around Barb. Barb was formerly the director of R&D Pharma GLP risk coordinator for GlaxoSmithKline in Pennsylvania and the UK. Based on her 42 years in the field and knowledge of toxicology processes and GLP regulations, and the history of why GLP regulations were written, she provides interpretation of difficult regulatory issues. Barb has been a member of the Society of Quality Assurance since 1991 and is currently a member of the board and is chair of the Specialty Section Council. Ms. Munch earned a bachelor's degree in science in animal science from the University of Maryland in College Park. As I said, we're very excited to have Barb share this wealth of information that she has around the regulations with all of you. And from now, I'll turn it over to Barb.
Okay, Karen, thank you. Just want to double check you can hear me well.
Yes.
Okay, great. So you might wonder, like, why is the history important to what we're doing? And the answer is really it's about the why. Karen knows I've always asked why. Why do we do something this way? And what I want you to understand, if you don't get anything out of this presentation except one point, is that each regulation in the GLPs is directly related to issues that were identified by the FDA. And this presentation only has a few because I'm trying to get through in about 40 minutes. Some of the examples I'm going to talk about are what happened at Searle in the contract lab Hazleton, industrial biotest, and biometric testing. Those are from the GLP side. I'm not intending at all to put any company or test article drug in a bad light. It really is to talk about training and how we get safe drugs to market. So, how far back do we go? I've only chosen to go back into the late 1950s. And this one example from then is the drug that's triperenol. This was the firm Merrill. The drug number was MER29. As a side note, MER32 you may have heard of, and another name is thalidomide. Triperenol was one of the first anti-cholesterol medications that came on the market, and I'm going to talk about 3 different species of their studies. Merrill had an interesting policy, which is that scientists were the experts. They could use their own judgment in deciding what findings to tell the FDA and which ones to not tell the FDA. For this NHP 16-month study that started in 1957, when in May the manager was working on reporting it, the technician made a graph of the body weights, and the manager demanded to the technician, you must remove one of the animals that showed test article effect and include instead a control animal from a different study, their body weights, to hide that evidence. On a rat study, there were only 8 high-dose females, and they all died or were sacrificed early. And because of that, obviously, there was no organ weights or blood analysis. In the submission to the FDA, they reported that only 4 of the 8 died early, and they had organ weights and blood values for all of the animals. Where'd they come from? From a 3-month rat study that wasn't this same test article. Finally, on this slide, there were increased lymphocytes on these 2 studies that the scientists decided were not significant, so they didn't report them to the FDA. Continuing on here with the dog study, there were only 5 treated dogs, and they all had some impact. 3 of them died early with liver degeneration, and 2 of them that lived had serious eye issues. When the Merrill told the FDA the report on this, what they said was there were simple liver issues and there were no eye issues. So the NDA was filed in July of '59 and approved in April of 1960. Right away, because it was one of the first cholesterol medicines, doctors were eager. They put it in patients. That wasn't necessarily the problem. One of the problems is that their sales reps, which you'll see are called detail men, these sales reps were very aggressive, and they had very strong advertising and sales. So they made a lot of money off of this medicine. In January of 61, this being one of the first cholesterol medicines, Merck says, I don't know if anybody from Merck is on the call, you guys were involved here. You synthesized the same chemical and you tested it in your own species there on site. And you discovered that it caused cataracts. And back then, there was a much more collegial relationship. So Merck called Merrill and said, have you ever seen this? And the Merrill personnel said, no, we've never seen that. Would you like to come to our site? Well, that will be lovely. So the Merrill people went to visit in February, saw exactly what Merck said, and saw exactly what they had seen in the animal studies. Neither company informed FDA. Now, it wasn't really Merck's job to do so. It certainly was Merrill's job to do so. They did not. And I'm sorry, wait. Okay, she's been pretty good up till now. Okay, so what was seen both in the non-clinical and the clinical was problems with hair loss, scaly skin, cataracts. This picture here is one I found in an article. This is from an 8-year-old boy who had high familial hypercholesterolemia, and he has a cataract there in one of his eyes. All those were seen in the animal studies, and they weren't reported. When they were reported, they were not relevant for MER29. Now, not only that, there were doctors and patients that called Merrill and said, have you seen any of these issues? And every single time the answer was, no, it's not related to our drug. It must be something else. So eventually, word got to the FDA. And in April of 62, the FDA came to Merrill and said, what have you got? Let's see all your data. And Merrill said, sure, here's all our data. And they matched it up to the NDA, and they found it didn't match. So they pulled all the data that they could from Merrill. And between the 9th and the 12th, the FDA then said to Merrill, you must withdraw your medicine or we will cancel it for you and we'll say you can't sell it. So this voluntarily, it wasn't voluntarily, they did it because the FDA demanded so. There was enough evidence for a federal grand jury to provide a 12-count indictment. In March of 64, then they pled no contest. They were fined $80,000, which seems like a lot back then, but they made hundreds of thousands of dollars, and this was a very small percent of their income. And their sentence was probation. So when you think about the stories that here, and I'm going to tell you, think about how they link into what you already know about drug development. And in this case, the concern is the company intentionally withheld negative information on both the non-clinical and clinical studies from the FDA, from the patients, and from the doctors. Right, so moving on, excuse me, I need to get some water here, to what is more commonly known is the information about the pharmaceutical company called Searle. And the trigger actually for the Searle investigation came from the drug called Flagyl. And in May of 72, Dr. Adrian Gross, who was a pathologist and a statistician, he was a brilliant man, he was asked to review 3 studies. They had stats, and they wanted to know if there was any indication of that. So between there, the 5th of May and the 17th of May, Adrian looked at not only those 3 studies, but also the NDA. And he said, well, hey, there's a problem here. Flagyl causes a highly significant increase in tumors of both rats and mice. Now, back in the 70s, there was generally a more collegial, open atmosphere between the agency and the firms. And so the FDA met with Searle and said, well, we don't understand, but Dr. Gross found these findings, and what do you think? And Searle goes, oh, thank you very much. We'll go back and we'll address these concerns and we'll run some new studies and we'll help and we'll tell you what we find. So in 1974, they submitted a number of documents, including the revised report from the NDA that they already had on file and that Adrian had already seen the first version. So now we have this revised version that Searle said they were just minor typographical errors. So Adrian said he had a great way with words. Instead of changing the summary to more accurately reflect the data, however, the data had been changed to more accurately reflect the summary, right? So he understood they changed the data. And the summary table was found to be in disharmony with the individual animal data. So the raw data didn't match what was in the report. So Adrian said, you know, we need to do something here. And he raised it up the flagpole, and people said, yeah, okay, so let's go have some more on-site visits at Searle. And those initial attempts to either visit Searle, or if they were allowed on site to review those records, they were unsuccessful. And that's a polite way of saying they were denied access. So this is the first example where I'm going to say you're going to link an event to a regulation. And I'm going to assume as we go through here, folks have seen these regulations, and I'm not going to read them word for word. But the point here is you always have to admit the FDA to come in and inspect your site. All right, so what exactly changed? Searle identified animals as, in this case, control male for CM number 21. There was a data change, and mind you, this is a control and a male animal. So in the 1970 report on page 59, it said fibroadenoma. In the report, same report on page 102, it was called an adenocarcinoma. In the 1974 report, they changed the first instance from fibroadenoma to an adenocarcinoma, which is malignant. So you'd think, gosh, it's really strange. Why would you change a benign tumor to a malignant tumor? What happened? Well, Searle later on admitted they didn't even look at the slide. They just changed it as a typographical error. Now, what they did was make it more serious. And the intent of that was to then have a control, especially a male, that had a malignant tumor, so it made the treated animals look less impactful. So this is linked to QA, who is going to match the reported results that accurately reflect the raw data of the non-clinical study. So this example is about raw data. Now, when Searle, there's a whole story about the Searle task force that did an investigation at Searle and at Hazleton Laboratories, and that was reported to Congress by Jerome Bressler, so we call it the Bressler report. Many of these links are in the slide deck, so if you're curious and you want to read the Bressler report, you've got access to it, at least the version that's public. So these raw data observations that weren't really defined as raw data at this point in time because there were no GLP regulations in place. They were documented by hand, and most of them were not signed or initialed, so you couldn't tell who did the work. And this is back just when Xerox copiers are becoming to be popular in use, and some of these records were copies with initials on them that were wet ink signatures. How did they get added on to copies, and why weren't they on the originals? And in this particular example, in housing group A, 26 of the 39 observations were copies with their original initials. So the FDA felt that they couldn't ever go back and reconstruct it. They couldn't find the originals. The observations indicated that this one particular animal, 23, housing group A, animal number 23, low dose male, was alive and then dead and then alive again and had some signs and then dead again. So we know animals don't come back to life. We don't know exactly what happened. And this is linked into 130E, where whatever there is, you have to know who did it and reason for change, sign and date. So this particular animal isn't on this next slide, but this is an example from one of the documents in the congressional hearing from aldactone. And you can see on the top left here, J24 high dose male was dead and alive and dead and alive and dead. And the one underneath was alive, maybe vanished, alive, and then vanished again. So what happened to the animals? Were they replaced? Would they jump cages? Were they not properly identified? The FDA was concerned because they couldn't track animals' status throughout the study. Moving on to study design and conduct, the FDA knew that there were 5 flagell protocols. They only found 1 from the 80-week rat study, and that wasn't followed. As an example, the protocol said this will be administered as 2 and 5% suspensions. However, 2 and 10% were actually used. And so if you look at it scientifically, if you say that a higher dose was used, then it appears safer than it actually was. They couldn't tie it into the drug dosage and administration to know that the animals received the amount of drug intended. So I said that's the first pillar of a GLP study. You must know which animals received what drug. And there weren't any administration records, or they were incomplete. This is linked into conduct of a study. You have to follow the protocol. Right. So many studies that were done at that time were called drug in diet. And that meant you didn't have to gavage, you didn't do any other kinds to get the drug into the animals. It was just easier to put it in their feed. And so what I have here is a picture of a Patterson-Kelly twin shell blender where you can put in the feed in the test article and mix it for a defined period of time. What the FDA found was there was no evidence of tests to determine blending characteristics or to validate the mixing time. And in fact, the inside was, in their words, encrusted with material from previous use. And mixers weren't grounded, which could allow the static of the test article to stick or not stick into the inside of the blender, which meant that not only was the diet contaminated from previous mixes, but your test article then could have gone on to future mixes. So you don't really know what was given to the animals. And now we know that in 5863, equipment is adequately inspected, cleaned, and maintained, and then generate, measure, and assess data. You have to make sure it's working properly. So this is an example here as a picture of a video that you are welcome to go to the SQA website and call it up. We have license to share it, that the FDA found that Searle didn't do any homogeneity tests on any batches, and they did 2 stabilities, but in those reports, it found that samples weren't homogeneous. So this is a picture from that video, which shows the test article lumps that the rats then could pick it out of their feed. So they didn't get the drug at all, they only got the diet. So this links into test facility management's responsibility for test and control articles being appropriately tested, and then 113, mixtures of articles with carriers. So now moving on into gross, meaning macroscopic at necropsy, histology, and then microscopic. There were approximately 30 animals that had substantial differences between what was reported by those who did that work on the path sheets when compared to what was submitted. And the examples on the next slide, so it all fit on one slide. I put here, we know for pathologists, the records of growth findings have to be available to them when they do their work, because they weren't. So in this example, for housing group A, number 2, controlled male, I'm not going to read this whole slide, don't worry, but what was submitted to the FDA was lung focal adhesion, 2 words, and when you look down there on the slide for lungs, there's 4 lines of description of what happened, what was found. And adrenal, moderately enlarged, and down below you can see it was also described to have spots, and then all other organs were unremarkable, and that's not the case because there were clearly some other tissues that have findings. So that's all I'm going to have time to say about Searle. I'll move on to Industrial Biotest. IBT was a contract lab, and it was one of the largest contract labs in the world at that time, and it did studies for virtually everyone in the industry, including the federal government, and they had a reputation of getting things done quickly and cheaply. They often underbid other labs. So how did that come up to the vision of the FDA? So what happened in 1975 is that Syntex was a firm that did studies at IBT, and the employee of Syntex had a whistleblower complaint and said, we've submitted naproxen to you, but there are problems with that. So Adrian had known that the study was done at IBT, so he pulled the IBT study. And he looked at that and he was totally surprised as a pathologist that there were unbelievably clean study data, and the rats on a 2-year study, no one developed cancer. Now, I'm not, for animals that were used on studies in the early 1970s and mid 70s, this is even, think, before cesarean derived, there were a lot of viruses and diseases going around, and animals died early, and animals had a huge number of tumors on CARC studies. So Adrian, as a pathologist, knew, of course, someone had to have cancers. So he says we need an inspection, and his quote here, what we found is enough to make your hair stand up, and we joke because Adrian was bald that he lost all of his hair because of this. So there was then a visit by the FDA to IBT in April of 1976. So there are a couple of stories here that have catchy names so you can remember them. One of them is called The Swamp. And if you think back in the dark ages of animals, a lot of them were in cages, and the only way to get water to them was hanging bottles. So filling bottles, hanging bottles, replacing bottles was a lot of work and a lot of manual labor. So Holte was the firm that had the first automatic watering system. It was designed to provide water and flush the waste from the cages, but it didn't work well. There were animals that got wet and had too much water, and there were some animals that had no water. Now, this swamp was a big room that had no floor drains. So there was at least 4 inches of water on the floor, and the technicians could only go in if they wore big rubber boots, like the kind you wear in a barn, the big heavy ones. There were a lot of animals that died because of the water issues. Either they had too much water or not enough water. And because the room was hot and humid, they decomposed quickly. And there's a quote from one of the articles: they lay in purple puddles on the dropping trays underneath. And because it was so humid in that room, technicians couldn't bring in their paper, so they had to write notes on the sleeves of their lab coats. So this is a picture from that video of the swamp. It's a very large room, and it held many, many racks of animals, including stacks of feed and bedding. And you can see there's no drains on the floor. So how does that link into what the regulations have? Animal care facilities, if you have animals, you have to manage the waste, and you have to have procedures in place to minimize vermin, odors, disease, and environmental contamination. In animal care, the FDA said clean and sanitize at appropriate intervals. We define what that is, and we put it in our SOPs, and then SOPs are required for animal care. So one of the test articles that was reported, investigated by the FDA, was TCC for the chemical firm Monsanto. They had an interesting philosophy of replacing animals. So this TCC study that had 3 treated groups, those 3 treated groups were in the swamp, and the control group was housed in a different room across the hallway, because the swamp was a very important room and it should have worked properly. So let's take care of the treated animals, and we'll just put the controls in a separate room and not bother with that space in that large room. Now, they had animals that died, so they ordered new rats from the breeder, and they called it a research study group. And anybody who died was replaced with a new animal. Technicians were forbidden to document it. It wasn't just that they chose not to. The management said never document it. It was never reported. Neither was the replacement of the animals. It was not in the final report. So what does it say in the protocol? In 120, you have to define the information about where you get your animals from, and it's linked into the study director, who's responsible for managing unforeseen circumstances. So certainly a bad watering system and animals dying are unforeseen circumstances that they have to be documented properly and take corrective action. One of my heroes here, besides Adrian Gross, is Donovan Gordon. He's a pathologist. He works at IBT, and he read the TCC study. Now, those animals that survived long enough to get any of the test article in them, He believed that all of the males that were treated had atrophy. There was a test article related effect. In November of '72, they went to FDA with Monsanto and talked about how wonderful this drug was. So the words here are he was told not to speak, but it actually is more sinister than that. I have it on good source, but not an official one, that on the flight out, they held him up next to the plane door and said if he said anything at all about it, when they returned, they would open the door and throw him out. So it's kind of serious there. So he didn't say anything at all. Between then and February '76, Monsanto said, you have to get Donovan to change his mind. We don't like these findings, and you have to find some other reason. There's a couple of examples here. Something else would have caused it. So Calandra, who's the president of the company, said, you must find a no effect level. So for 3 years and 3 months, Dr. Gordon was asked to change his mind. At the end, Dr. Calandra, the president, edited Donovan's report and removed them and said autolysis precluding meaningful evaluation, meaning none of the testees had any diagnosis at all. And then he puts it on the desk and says to Donovan, now sign it. So he was forced to sign it or he was going to lose his job. And here's the quote from the trial. He didn't want to leave IBT, so he signed the report. So this is one of the reasons that we have individual scientist reports and we have pathologists who have special exemption because they're the ones who fill the second pillar of a study, what actually happened to the animals. So you can link the test article to the animals. Moving on to the magic pencil study, another one of those fun words. It's called magic pencil because pencils were used to generate data out of thin air. So the technical writer, his name was Phil Smith. He went to find the blood and urine data in ClinPath, and they couldn't find any data. So in his handwritten draft, those tables were blank because they couldn't find any data. So Phil gives it to his manager and he says, I found that these tests weren't done, and this study as a whole is so bad, I don't want to sign the report. And his manager goes, oh, of course, fine, don't worry about it, I'll take care of it. So he didn't think anything of it, went to the typing pool, and finally, oh, look what came back from the typing pool was the final report. And in that final report, not only were there reported blood and urine values from samples that were never collected, but his signature was there. Now, it wasn't like they falsified his signature. What they did was they took a previous report that he had signed, made a Xerox copy of that, cut out his signature, pasted it on this new study, and then submitted it into the agency. So he knew when he looked back at the tables that were blank and had writing in pencil, that was his boss and his boss's boss. So here's an example from the video that shows the values from that filled in form, and actually they look pretty good. So, you know, if you're going to make up values, you're going to make up good values. So that links into the regulations on the magic pencil study, right? What's really important is not necessarily what's in 130 about recording, but one that's in the definition in 3K, which is important for reconstruction. Because as we get our studies done, it's not just that we know what happened, it's that can someone in the future, especially the FDA, reconstruct the work that was done. It's linked into the protocol. You have to know what type and frequency of tests are going to be done. And the study director has to make sure that the protocol is approved and then followed. So if you ever wonder why sometimes the wording in the regulation seems odd, like of course you would have a protocol and of course you would follow it. Well, it's because they didn't do it then. Moving on to biometric testing. This was located in New Jersey, and in this firm, they actually didn't have sponsors come on site. They fabricated their data, whatever it was that they were working on. They said to your sponsor, the sponsors, you mail us your test article, give us the protocol, you know, the information, we'll run your studies and we'll give you a report. You don't even have to come here on site. So the sponsors thought, this is great. So they just submitted to the FDA. So they had no knowledge that there was anything wrong going on until the FDA came on site and found a few examples. Technicians weren't trained, They didn't carry out the lab tests. They twice falsely claimed to have tested 2 drugs in 70 animals. That didn't happen. In 1979, 5 officers were indicted, 2 folks pled guilty, and they stopped conducting non-clinical studies. So that is linked to 5810, that a sponsor who utilizes a contract lab has to notify it the service is part of a study. And not only that, there's no longer the ability for sponsors to say, we didn't know. So we have a stronger emphasis now on study sponsor monitors, because the sponsors can't say, I didn't know that the lab was doing something wrong. So in one of the congressional hearings, now this is between the Congress hearing what the FDA reported and what Searle said. So at the end of the reporting, Ted Kennedy says, and these are the excuses that Searle made: inaccurate science, sloppy science, fraudulent science. These are the greatest threats to the health and safety of the American people. Whether the science is wrong because of clerical error, as they claimed, or poor technique, or incompetence, or negligence is less important than the fact that it's wrong. So Kennedy got it right. Kennedy was friends with Adrian Gross, and that helped certainly getting this raised up into Congress's awareness and how it led into the GLP regulations. But as a statement to that, what Ted Kennedy says, Daniel Searle, who was Daniel Searle of the Searle Company, he was one of the presidents, he says, in the truest sense, the errors identified by the FDA, the deaths and masses, were completely irrelevant to the scientific conclusions of the study. Now, I can't defend that. I don't know how a person could stand up with a straight face or put in a statement and say those errors were completely irrelevant to the scientific conclusions of the study, because we know today, of course, those are relevant. Right, so I'm going to shift now from the GLP to the GMP. Now, I am not a GMP expert, and I don't know a whole lot about the GMP history, but I can report this this is from an article from the FDA historian that I've had some contact with for the GLP work. So this firm was called Winthrop. And think about what was going on in the 1940s. After the Depression, there wasn't a whole lot of antibiotics available. Now we're getting ready to go into war. It's a hard time. So they need to get some medicines on the market. So they work together. So Merck, Squibb, Maltby, Calco, Lederle, and Winthrop all worked together, and they had the first 6 antibiotic NDAs approved. And that was in July. So now just focusing on Winthrop, the 22nd of December, a Winthrop detail man. Now that's what we call now the sales reps. They called them way back then detail men, because they were, well, men. He reported back to his headquarters that in Kentucky, 12 patients had adverse reactions to sulfathiazole, and some of them were unconscious. Now mind, sulfathiazole is an antibiotic, right? So on the 24th, so they got right on it. In 2 days, their analysts confirmed that of lot MP29, 20% of those tablets were contaminated with barbiturate. That was their brand of the drug luminol is their brand. So they knew that they were contaminated. So 2 days later, like mine, Christmas is right in there, they said, right, so let's go back and we're going to tell all of our detail men, we want you to collect and return just the MP series lots, but especially MP29, because the tablets don't disintegrate properly. So they didn't give them a real reason. They didn't give them the truth. They just said they didn't disintegrate. So I don't know how much urgency there would be for that. It told the detailers to contract the wholesalers and the hospitals and those retailers that were important. But you don't need to talk to doctors or patients or media. Don't tell them. And oh, by the way, don't tell the FDA. So between when it went on the market and this time in December, there were 18 deaths and 57 adverse reactions that were linked to this barbiturate. Between that recall and the end of March, there were at least 64 more deaths and 143 adverse reactions. In March of 1941, there was a 3-year-old girl in Boston who had an infection, and she was put on this sulfathiazole. She went into a coma. She did recover. So she took 2 tablets, which was 15 grains. Her father knew somebody at the Massachusetts Department of Public Health. So he called up his friend and said, look, my daughter just took this medicine. This is bad. So the person at the Department of Health then contacted the Boston FDA office and said, hey, we have something going on. And this was the 19th of March. So the FDA went right on it. And they went to Winthrop. There were a number of investigators on site, and they reviewed it. So you folks who are GMP experts, not me, would be able to then link this to how the GMP regulations are written. So what they found was that the contamination probably happened during the tableting. These contaminated tablets were almost entirely luminol, meaning there wasn't any sulfathiazole there, but it was mostly the barbiturate. The tableting machines for those two were adjacent to each other in the same room, and they were used interchangeably. So it could be one, could be the other. So there's a way that they could have been contaminated there during the tableting. And then finally, the last clue there is that this manager of the tablet production area said that one of his employees, it might have been linked to that one person. He had a history of being careless, and he had other problems, which you got to wonder what that means. But there were some other factors in there, and that person was let go at the end of December anyway. So they're hoping that that's the end of it. So we come into recalls now. They're back to the previous slide, right? They only got some of the medicine back, though the FDA tried really hard. Once they knew, they escalated it up. So Winthrop said to their sales reps, oh, by the way, you need more. You need more. You need to go find some more. Well, finally, the FDA said, we're tired of you guys playing games. And they issued a press release to the public that said, now everybody needs to know what's going on. And then not long after that, Winthrop said, okay, I guess we better notify the pharmacists and the physicians and the drug wholesalers and hospitals and medical societies as a way to get the word out. Most of them had never heard of not only the recall, but the real reason, the fact that it was contaminated with a barbiturate instead of an antibiotic. So as a summary of what they found, the FDA found about 29% of the mother lot of MP29 was contaminated. Those tablets typically had no sulfathiazole and 350 milligrams of phenobarbital and the excipient. Now, the typical phenobarbital dose was 100 to 150 milligrams, and these tablets had 350 milligrams. Sadly, about 100,000 tablets were never recovered, most of them from that lot, so they don't know what happened with them. So in April of 1941, Winthrop was the first firm, woohoo, not for them, to have their NDA suspended because of lack of proper manufacturing controls and actions that were inconsistent with controls that were indicated in the NDA. So they said they were going to do things and they didn't do them. Winthrop refiled the NDA in May, and then June revoked the suspension because they identified the issues and they wanted this drug on the market to help with the war and to help patients. So you can see a picture of the box there from sulfathiazole. They charged Winthrop under the Federal Food, Drug, and Cosmetic Act for selling adulterated and misbranded sulfathiazole, and Winthrop pled guilty. In the end, nearly 300 Americans were killed or injured, and they had a whopping fine of $15,800, which isn't a whole lot. So this slide has the links to, if you're interested in the World of Action video from 1984, there's a couple of articles here and the Bressler report link, and then a link to the sulfathiazole disaster. So a couple of more slides and then I'll be finished here. The future state of FDA GLP compliance. The FDA has changed, effective the 1st of July, from remote regulatory assessments, which weren't an inspection because they could not show their 482 and they did not write 483s. They're now back to on-site inspections, in person as necessary is the official wording, because some of them are still doing remote regulatory assessments as they need to, but they're really working hard to get caught back up. The status of the GLP final rule, the new regulations that will be coming, we have no idea when. Keep in mind that this review started in 2007. The ANPRM was December 2010. There were over 3,000 comments that were received. And because of the rulemaking process, they had to address all of those comments and put them in the preamble. Then we got the NPRM. They received 78 submissions with hundreds of comments, and they've updated their documents. It's still in internal review for staff comments. It has not passed through final agency clearance, the rest of it through the rest of the agencies. And a tip from Barb, don't change any processes based on the NPRM. I audited a lab not long ago that was working to the NPRM. And I said, no, you have to follow current regulations. You can't change till the NPR comes. Now, what we heard for a couple of years is that there'll be a 12-month period for compliance. Eric Pittman said earlier this week when I taught a class with him that it could be a year or two for compliance. So stay tuned. And then what's keen, especially for pathologists, is that the draft peer review guidance, and that still is undergoing review, and we have no idea when that one's coming out. Because I'm a OECD GLP discussion group member, I can give you notes from the official notes from then. That was the meeting in Sendai in Japan in April of 2021. So we have the draft document regarding data integrity. Now, that one actually was issued on Monday. So we have that one. If you don't have a copy, just holler and we'll make sure you get it. There's the 2020 stakeholder review where they asked for emerging technology information. And there's a number of types there. They are working on a cloud-based system document as we speak. And they're also working on a separate document that has other new technologies, more generically, how to address them. Finally, there's the draft revised document on quality assurance and GLP. And as we know, that was also issued this week for comment. Comments go back to our US contacts, which is Eric Pittman for FDA and Francis Leem and Dan Myers for the EPA. So if you are interested in commenting on it, SQA is managing that response, as well as you can go to the public website and answer those topics. So I wanted to point out that what I presented on the history is part of what I know. I'm working on a book on the history behind the GLPs. There's a Quality College Class 102 that will be next Tuesday, and it's not too late to sign up. If you're interested, find it on the SQA website. There's my contact information in case you have any questions. And then I'll close. If we don't remember the past, we're going to repeat it. So I'll work with Karen and Chris. If there are any questions that you would like to discuss, I'm happy to. We have about 14 minutes left. So I'll turn it over to you and Chris, Karen and Chris.
Okay, thank you very much. We really appreciate your insight there, Barb. It looks like we have one question in the questions bar. I'll read it to you and you can follow along. I'm sure you can probably see this. Could you share your interpretation of the GLP regs as related to the GLP QAU responsibilities to conduct study, protocol, and final report inspections? Given the shift in industry and regulatory emphasis on risk-based audit programs, do you feel that it would be acceptable under the GLP regulations for GLP QAUs to audit representative samples of, sorry, I lost my step here. Acceptable, boy, this keeps shifting on me. For GLPs to audit representative samples of protocol and reports, i.e. process inspections, instead of inspecting protocol and reports on an individual study basis?
That's a very long question. Yeah. And I'm going to say that the US philosophy has not changed. And this is Barb's opinion. I don't speak for the FDA and never worked for the FDA, but because of what happened in the US, and it basically never happened anywhere in any other countries. There was an investigation, but they never found anybody that was bad players like in the US. So the FDA has GLPs, and we are required to follow US law. OECD, we are an OECD member country, and we have a mutual acceptance of data, which means we will accept data for review from other member countries, and they will accept data from our country. But we are following US law, and US law is not going to change significant. So first of all, follow US law today. So if you're talking about how much could you stop auditing, I would say that's not an FDA recommendation. So if you think about some of the stories that I said, where there was no oversight, there was no independent auditor, when they found all these issues and they worked towards developing the draft GLPs, the FDA looked at the GMPs as an example, but they said, we need more than different from what the GMP regulations have. Because in our world, we need a single point of control, which is the study director, who's in charge of the study. We need test facility management, who's responsible for the facility and all the supporting work to get the studies done. And we need someone who is, as I learned in 1979, the FDA's left hand. As I've heard the FDA, Nicole Victoria spoke recently, and she used the words as FDA's proxy. So the FDA thinks so highly of the QA role to follow up and be independent that they set them to be able to say whatever they wanted, and their audits are shielded from routine FDA inspections. So the long answer, I think I've answered your question about interpretation of the regulations, QA responsibilities. Based on the few examples that you've seen, there was no independent person who said, hey, no, you can't do that in the industry. So I hope I've answered that question. Chris or Karen, any feedback on that?
Right, so the final report is actually required, right, Karen? So, or sorry, Barb, so that is something that I would see you would not be able to take a risk-based approach at, because all final reports need to be audited.
Right, so you, correct, that would not be something you could not do as required by the regulations. So where it's appropriate to put in a risk base is to say, like you heard me say, there are two pillars of a GLP study: test article, getting applied to the test system, and then pathologist knowing what happened. So if you were going to risk base, you'd say those are the two most risky areas I want to focus on. Those are the two most important parts of a study. But you also need to look periodically at clinical pathology and fill in the blank. I would not change anything based on a risk-based approach that turns away from meeting the GLP requirements. And I would be surprised if there is much of a pullback in the new GLPs away from the role of quality assurance as defined in the GLP side.
Right, thanks for that. It doesn't look like there's any other questions, but along that line, talking about the new regulations, do you feel that the shift now to executive management as described within those, you know, the NPRM, that FDA will move to institute that? Because I know there was a lot of pushback about, you know, all the way up to CEO and others of, you know, responsible for the study level type oversight. What do you feel the move toward that's going to be, especially in you know, looking in light of large companies, big pharma, let's say?
Yeah, I have had no insight as much as I have asked. We have asked through SQA to say, let's partner, let's help you. You know, we're in this together. They know that, but they are prohibited from inviting us in to know what's going on and even to share. and in part they can't share because it could change, and they could say, well, you know, Dr. Cochran said from the stage this in this year, but now it's changed. So the part about management, I think, is going to be more relative. So when I started in 1979, back in the dark ages at Hazleton, we were a firm then of maybe 200 people, and it was very easy to say, here's QA, here's management. There were only a few people there. But now, and I'll pick on my former employer, GSK, it is 100,000 people. And who really is test facility management? Is it the CEO? Is it further down in the line? And where does QA come in to report and fill in the blank? So really, in my interpretation of what they're looking for from a test facility management or whatever it's called, is that the person that has the ultimate accountability and control over the budget and hiring and selecting work to be done. So there, in big companies, there's going to have to be some benefit. Some benefit isn't a word. Sorry, some connection between like the president of a firm that has 100,000 employees will not understand GLPs and is not the right person to answer the questions. So I'm not sure I can really answer your question there.
Yeah, no, I agree. I think, you know, it's interesting to see that line of requirements that they're putting out because of the different environments that we deal with. And like you said, some of those higher management folks definitely don't understand GLP on the base level. And it would be a shame for them to, I guess, you know, be, you know, pulled into and try to answer questions related to the GLP study. We're already requested, you know, for organizational charts up to the CEO and those types of things, but yeah, have not been asked to pull a CEO into a FDA inspection yet. So let's hope that doesn't.
But they could, if it was a serious enough event, they could go that high. They have the legal responsibility to do, you know, authority to do so.
Okay, Karen, would you want a question? We have a couple questions that we had aside here. I can go to those.
It looks like there's a couple more in the Q&A. Is there a specific Part 58 reg which requires the GLP QA audit each study protocol?
So I'm looking at the regulations as we speak, because while I used to have them memorized, I don't have them memorized anymore. And Karen, please step in because you are still actively in QA. There is a difference between, there's specific wording in the regulations that says here is what you must do, and the actions that are required by QA to assure themselves of the work that's being conducted. And so I can't, off the top of my head, I can't find it right now that says they must specifically audit a study protocol, but I know that it is something that's important, because if the study protocol itself is not compliant with the GLPs, it's QA's job to find that and let the study director know that there are compliance issues. Karen, can you add anything to that?
Yeah, I'm actually looking in mine too, but I think the whole thing about the review of the protocol came out with the OECDs. And so a lot of the companies that say that they're following the FDA GLPs are also saying they're doing the OECD. So that's where a lot of that push came in, you know, years and years ago for QA to review the protocol. But you know, I think it is kind of vague in the FDA GLPs at this time around specifically for the QA to audit the protocol.
There could be more specific wording in the preambles. I don't know.
Right, and you know, with the multi-site study process that's so prevalent these days, you have different QAUs either performing a protocol inspection of the specific parts that are being conducted at their site, but yes, we've seen that there are areas where it's reviewed at, let's say, the first inspection that's done, or it could be conducted later on during the study too. So yeah, different companies take a different tack toward the protocol.
So a
Yeah,
non
and somebody just put in the Q&A around the actual regulation, and I think that's the thing. The key word is adequate, and what are you finding is adequate to ensure the integrity of the study? And as you said, Chris, other companies have different interpretations of that.
Okay, we've got about a minute left. Anyone else want to ask a quick? We have Barb's contact information. If you think of one and want to follow up, we can always send that and get an answer for you at a later time.
certainly.
Well, thanks again, Barb, for your time, and we really appreciate your insight on this. And hopefully we could maybe do this again sometime.
Yeah, well, I've got lots more that I could share. So you guys just let me know, and we'll figure out something. This topic, or OECD topics, or fill in the blank. So, and thank you for the invitation. I appreciate it. I'm always happy to talk about GLP history.
Great, thank you, Barb.
Yep.
Have a good afternoon, everybody.